So the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series will include different research areas in molecular biosciences, as well as concessions supports your career development. And each webinar will give you the opportunity to ask questions via text, and we welcome questions for future topics and speakers to our future webinar series. So please see the details at the end of this webinar again. So I'm Vanja Pekovic-Vaughan. I'm in musculoskeletal physiology at the Department of Musculoskeletal and Aging Science at the University of Liverpool. So within the Biochemical Society, I'm a committee member and a vice chair of research area 6, biological systems. And I'm also one of the network coordinators of the ECMage network, which is one of the 11 UKRI-funded interdisciplinary networks that bring together expertise in key aspects of aging, biology, chronobiology, computational modeling, engineering across Europe. And we offer free membership and pump priming and exchange funding to support a variety of projects. So our speakers today are members of ECMage network, and I'm delighted to share today's webinar entitled Extracellular Matrix Aging, Forging New Links and Development of Cutting-Edge Models and Methodologies, which is part of the Biochemistry Focus seminar series, a dedicated early career research program of webinars. So we are delighted to provide this opportunity today for Anna, Milos and Daniel, who will share their novel work with the molecular biosciences community. So before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, so please send your questions during the talks. If you have a question, just type it in a question box as shown in the image on the screen and state who your question is for, and we will answer as many questions as possible at the end of the webinar. So our first invited speaker today is Anna Hoyle from the University of Oxford. Anna completed PhD at the University of Manchester in Joe Swift's lab using heavy isotope labeled proteomics to study matrix turnover. She is now a postdoc in Kim Midwood's lab at the Kennedy Institute of Rheumatology at the University of Oxford. And her research currently focuses on studying matrix turnover in arthritis, as well as collaborating with many labs across the Institute to advance proteomics research. So today, Anna will share her work on quantification of protein turnover by isotope-labeled proteomics that identifies pools of extracellular matrix with distinct dynamics and differential responses to aging. Over to you, Anna.
Okay, yes, so I'm talking about using heavy isotope-labeled proteomics to study turnover, particularly of the extracellular matrix, and how that changes during aging. So the reason we're studying turnover is because the amount of matrix you have is important, and so too much or too little causes issues, and so really that's dictated by how quickly or slowly your matrix is turned over. And so the tendon extracellular matrix was initially really thought to be pretty static and barely turn over at all, get secreted during development, and then that's it pretty much for life. And this study here showed that. So they used the fact that there's a peak in carbon-14 in the atmosphere around the same time as nuclear bomb testing in the early 1960s. And it could show that if you're developing during that time, you incorporate this heavy label into your tendon tissue, but if you're not, then you don't. And you can still detect this peak sort of 40 years later when they detect it. The problem with this model, though, is that it doesn't take into account how that tissue doesn't fail. So we know that your tendon experiences daily stresses and strains from mechanical loading, and we know that's associated with a certain amount of damage. And so how does that not sort of build up over time and eventually cause the tissue to fail? And Joan Chang's lab and the Kata lab showed that there was actually a circadian pool of collagen which might explain this. So these smallest collagen fibrils were found to be circadian, and they predicted that this pool was kind of protective and it absorbed the daily stresses and strains that the tissue experienced and kind of was sacrificial. So because it's turned over much quicker, these damage doesn't accumulate and it therefore protects your more permanent matrix. So they took tendon tissue, and to try and show this, they extracted the circadian fragments. So they did a mild detergent wash, and that solubilized these tiny, smallest fibrils that they found were circadian. They took the remaining pellet and they did harsher solubilization on that to get this F2 fraction. And when they did sort of a time course proteomics, they could see that collagen 1 in the circadian pool was nice and rhythmic, but it wasn't in this more permanent pool. And our aim was to kind of build on this, but to really show that they had two distinct rates of turnover between these two fractions. And to do that, we used SILAC. So this is using a heavy amino acid label. In our case, we used carbon-13 and lysine. So here, all of the carbon atoms are switched out for carbon-13, which leads to an increase in mass of 6 Daltons, and that's detectable by mass spec. And so if you feed the mice this heavy label, over time, that amino acid is incorporated into its proteins, and so your protein population becomes sort of labeled over time. And a faster protein will be turned over quicker, and so it incorporates more of the label and becomes labeled faster than a slow turnover protein. And by monitoring the decrease in light protein and increase in heavy protein, you can get an idea of the turnover of the protein. So what we did is fed mice this heavy amino acid diet for 1, 2, 4, or 8 weeks. And you can see on the right here, we see this really clear increase in the label as we feed them for longer. We then take those heavy to light ratios and convert them into half-lives. We use a method already published up here to do this. And essentially it uses the two pools. So you have a soluble lysine pool and you have lysine that's already incorporated into proteins. And the complication really comes from the fact that the diet that we can control feeds into the soluble pool, but so does amino acids from protein degradation. And this degradation is going to start off being degrading light protein and over time start to degrade heavier protein. But using this model, we were able to calculate half-lives for over 1,000 proteins in the tendon with an average about 15 days. And if we group these half-lives into different groups and then do gene ontology on that, we can see that these fastest turnover proteins, so ones with half-lives of 0 to 10 days, are enriched for the immune response and inflammation, protein folding and transport. Then as you go up a bit to 10 to 20 days, we see translation, 20 to 30 becomes metabolism. And then as we get slower turnover, we see things like cell adhesion, chromatin, and in the very slowest group, we see the matrix. So going back to our initial aim, we took our tendon tissue and we fractionated it into this F1 pool that contains the circadian collagen, and an F2 pool, which is more permanent, that contains non-circadian collagen. And initially, these two fractions are pretty different. So the F1 is a much more intracellular, and the F2 is much more matrix heavy. And if you look at that matrix particularly, F2 is largely collagen, whereas F1 is a bit more it's got more other things going on. So it's got more glycoproteins and proteoglycans. And if you look at the turnover of these two pools, you can see that F1 turns over, or the matrix in F1 turns over much quicker than in F2. So higher heavy to light ratios is faster turnover. So the blue line is much faster turnover. And if you look specifically at collagen 1, we see that same thing. So we see much faster turnover of collagen 1 in this F1 pool compared to F2. So we could kind of separate out these two pools of matrix turnover and these two pools of collagen 1 specifically and show distinct rates of turnover. So here again, we see these significantly different half-lives between F1 and F2 in our adult mice. And if we look at the matrix groups, we can see slower turnover of collagens compared to glycoproteins and proteoglycans. There's quite a lot of distinctions between these different matrix groups. And what we wanted to know is whether this changed as the mouse ages. So we took 8-week, 22-week, 52-week, and 72-week mice, and we fed them for 4-week pulses on the same diet as before. And you can see that generally the heavy to light ratios decrease with age, so you get slower turnover in aging. And more specifically looking at this matrix and these fractions again, in the young mice, we see no separation between this F1 and F2 pools. And you can see hopefully that that's largely because this F2 fraction isn't as slow turnover as it is in adult. So we think it's because it's not sort of properly laid down its permanent matrix. Aging, we also lose this separation between our fractions, and that's largely the opposite. So F1 is becoming slower turnover in both of these cases. And we also lose some of these distinctions between our matrix groups in aging as well. Looking at individual proteins, this is comparing our adult mice to old and aged in F1 and F2. And so these red proteins are all proteins that are slower turnover in aging. You You can see that in F1 fraction, there's quite a bit more going on, or more proteins being significantly slower turnover in aging compared to in F2. And also in collagen 1 specifically, we see there's a really clear decrease in turnover of collagen 1 in F1 versus, it's still a decrease in F2, but not as clear. I looked to see why that was, why we might be seeing these bigger changes in F1. And in terms of abundance in the sort of more general proteome, we see decreases in translation and also decreases in the matrix as well with aging. And then also in turnover, so these are all pathways enriched for in slower turnover proteins. We see things like cell adhesion and response to mechanical stimulus, which have been reported to happen in aging. We also looked at circadian matrix proteins. So these were previously identified from a different study, and we see that these in adult mice, the circadian matrix proteins are significantly faster turnover than non-circadian. But actually in aging, we lose this distinction, which fits with sort of previous studies that have shown dampened circadian rhythms in aging. We seem to see this. The F2 more permanent matrix is more consistent in aging, but it does change a little bit. So this is looking at the most slowly turned over proteins that we can identify. So this is what we've termed a kind of permanent matrix in adult mice. And then if you add proteins that also fit into this category in old mice and in aged mice, you can see we start to include a few more collagens and also some basement membrane components as well. Going back to our initial kind of hypothesis, we thought that this circadian pool was acting to protect this more permanent one. So if we're seeing this slower turnover and reduced abundance of this F1 faster turnover circadian fraction, are we seeing an increase of damage as a result of that? And we can see increases in post-translational modifications with age here. We also see increase of semi-tryptic peptides, which are indicative of degradation. We see that trend happening for the general matrixome, but for collagen 1, we see this really significant difference in this, a substantial increase in the amount of degradation happening with age. So in summary, in our homeostasis, we see this large separation between F1 and F2 fractions. We also see distinctions between different matrix groups. But then in aging, we lose this distinction. We don't see so much separation between our fractions, largely due to a slower turnover of F1. We also see general decreased protein turnover, dampened circadian matrix turnover, loss of proteostasis, or which might be kind of factoring into this slower turnover of F1. And we think as a result of that, we see this accumulation of matrix damage. So yeah, thank you for listening, and thank you to all the people that helped with the project.
Thank you, Anna, for that really interesting work. We will now have our second speaker, which is Milos Marinkovic from Indiana University, and we'll leave any questions to yourself at the end of the webinar. So Milos is an assistant professor in the Department of Orthopedic Surgery, and he's also a member of Indiana Center for Musculoskeletal Health, and also a research health scientist within Richard Rudesh Medical Center in Indianapolis. His research focuses on cell matrix interactions in bone marrow progenitor niche and the effects of aging on extracellular matrix. And today, Milos will discuss his work on CCN1, a key metrocellular regulator of skeletal MSCs, is depleted in aging bone marrow stromal niche. Over to you, Milos.
Thanks, Vanja, and thanks everybody. It's very nice to be here, and it's a real pleasure to speak to everybody. So just to give you kind of a walkthrough of the approach we've taken. So we basically make extracellular matrix in vitro from primary human cells, and we've used this method to make extracellular matrices that are specific to the tissues the cells come from. We published, extensively over the last 10 years or so on different tissues and how the matrix made by the cells from those tissues actually aligns with the tissue, the native tissue, and how it looks at cell behavior. But aside from tissue-specific effects, something that I've interested in, and I've now established my own lab at Indiana to do, is to look at aging using this method. So what we've done is taken young bone and aging bone from human donors, and we've isolated MSCs from that bone. And we produced extracellular matrix. And then we looked at the extracellular matrix and first tried to characterize how is it different? Is it different physically? So we saw that young matrix, or I would say matrix made by young cells, and I'm going to refer to that as YECM. And then the one made by aging cells, I'll refer to as AECM. They have very different organization. Young matrix has these very fine, highly parallel aligned fibers, and the aging matrix has these highly bundled, relatively disorganized fibers, and you see their alignment is much more broad. So they're not all sort of going in the same general direction. Then we looked at the mechanical properties. As you can imagine, the structure of these different matrices also translates into very different mechanical properties. So aside from roughness that is characterized by the actual physical roughness of the surface, you see that the storage modulus of the matrices is very different. So the young matrix is actually stiffer than the aging one. And that's actually interesting because the stiffness range of the aging matrix made by aging bone marrow MSCs actually is much closer to the range of the matrix made by adipose MSCs. Young matrix is much stiffer. Now we looked at whether those young and aging matrices actually direct fundamental lineages of MSCs, so bone versus fat, and if they differently direct their differentiation. So here we stained for calcium, and we cultured cells in osteoblasts or adipocyte media. The adipocyte media we stained for lipids, and we looked at the deposition. So we saw that young MSCs on young matrix had much more calcium deposition, so kind of a surrogate of osteoblast differentiation, than on aging matrix. And we found that on the aging MSCs, when they're on young matrix, actually deposited more calcium than aging cells on aging matrix. Actually, that condition deposited the least calcium. Now, when we looked at lipids, we saw sort of the opposite, where young cells on young matrix, culture and adipocyte media, were actually relatively resistant to differentiation to the depositic lineage. That young cells on the young matrix, on the aging matrix rather, had more lipid deposition, and then we saw the aging cells on the aging matrix actually had the most. And what's curious about that is it suggests that really these different matrices have very different signaling properties and collections of cues that sort of take cells into different tissue-specific lineages. So the question is, well, how are they different? So we of course looked at proteomics, and we saw that they have markedly different collections of proteins that are in there, and different proportions of collagens and proteoglycans, just as classes of extracellular matrix proteins. But we actually looked at the matrisome. We saw that the aging matrix compositionally was much more diverse than the young matrix. And curiously, there were some proteins that were actually specific to the young matrix that we didn't find in the aging matrix. And the most abundant of those was a protein called CCN1. Again, there's others, but we started with CCN1, one, because it was the most abundant, and also because we saw, looking at literature, that there was some interesting work that was done around the time we started relating to this protein and how it directs cell responsiveness to bone-specific growth factors. So the CCN family it's a family of 6 different CCN proteins, and CCN1 is of course one of them. What's interesting is that they have these different subunits that all have binding sites for very important cell receptors or matrix proteins. So, just structurally, it seems to imply that these proteins, this class of proteins, is sort of an adapter that facilitates cell matrix interaction. So we have subunits that bind to integrins, we have subunits that bind directly to growth factors, and then we have subunits that actually bind to matrix proteins. So it's a protein that interacts with cells, it's a protein that interacts with growth factors, and it's a protein that interacts with matrix. How does it influence cell behavior? So the first thing we did was to actually see well, what happens if we take this protein, a recombinant version of this protein, and just add it exogenously to cells cultured on plastic, and we'll compare that to cells just cultured on young ECM, and we'll look at proliferation. And we saw that the exogenous protein did increase the proliferation somewhat, but no concentration or no dosage that we tested actually was able to make equal the proliferation or the cell density that we achieved on young matrix. We also looked at BMP2 responsiveness. I showed that it's been suggested that CCN1, or also called CR61, binds to BMP2. So we looked at, okay, well, how do cells behave when they're treated with BMP2 on the different matrices, and how do they behave when we supplement this protein? So we looked at BMP2 responsiveness, and we measured RUNX2 as a sort of measure of responsiveness to BMP2. And we saw that on plastic, with or without the addition of this protein, cells were not responsive to BMP2. On young matrix, they showed a significant upregulation in RUNX2 when we treated them with BMP2. On aging or elderly matrix, we saw that there was no responsiveness. And when we supplemented the protein to the aging matrix, we similarly saw no responsiveness. So I should clarify here that these are actually very low dosages of BMP2. We found in our previous work that culture on matrix allows cells to respond to much lower dosages of BMP2 than normal. So we've already titrated these to be very, very low dosages where you see a response on matrix, but you don't see a response on plastic. And we think that this actually much more closely, mirrors the physiological situation of how much BMP2 cells are going to see in the body. So we said, okay, if adding this exogenous protein doesn't recover the function, well, how can we actually see if it has a role that's matrix specific? How can we better study the matrix specific role? Because again, we've shown that this protein is one of the factors that is very different between young and aging matrix. So we created a young matrix where we used siRNA to block the incorporation of the protein as the matrix is being made. And then we created this young, aging or elderly, sorry, some places I switched nomenclature at one point. So it went from ECM for elderly matrix to AECM for aging, but it's referring to the same matrix product. Then we have an aging matrix that has relatively little CR61 or CCN1, but then we increased its incorporation by using an adenoviral vector. So we basically created a young matrix with very little CCN1 and an aging matrix with quite a bit. And then we looked at BMP2 responsiveness, and we saw something very interesting. So we saw that the young matrix, of course, we see very healthy BMP2 responsiveness. However, on the elderly or aging matrix, when we restored the incorporation of this protein, we're able to really increase the responsiveness to BMP2, which was exciting because it suggested to us that there is a matrix-specific role for this protein in how cells respond to BMP2, and that you really need this protein to be integrated into the matrix to see this effect. And of course, there was kind of another interesting translational angle there, which was that, well, if adding this matrix component to an aging matrix can really increase the sensitivity of cells to BMP2, then this may be an interesting orthobiologic or be useful clinically, therapeutically. And it's kind of made us think more about matrix engineering as a way to sort of counteract some aging effects in tissue. We also looked at IGF-1 responsiveness because there is a putative IGF binding site on CCN1. So we used these same matrices and we treated cells with IGF-1. And we saw that actually by restoring the incorporation of CCN1 in the aging matrix, elderly matrix, we're able to, just like with BMP2, restore responsiveness of the cells to this protein. So then we looked at the tissue, and then we said, okay, well, if we see this on a matrix level, what can we infer about the situation in tissue? So we first went and looked at, we tried to define an age range where there was a really significant drop in bone mineral density in mice. And, we actually looked at literature and we saw, we saw that there was, big consensus that bone mineral density in aging mice goes down. But when we actually looked at aging mice that were, you know, in the 18-month range, we saw that there still really wasn't that marked of a difference. It really took us going into after 20 months, so 21, and we looked at mice all the way to 33 months old, where we saw a really consistent drop in bone mineral density, and we were looking at vertebral bone mineral density specifically. So once we defined those age ranges, we actually harvested the tissue and we decellularized it, decalcified it, and then we actually digested it and looked for CCN1 in the tissue matrix itself of those mice. And actually we saw in the young mice, there was plenty of CCN1. And then when we looked at the lumbar spines of the aging mice, we saw a significant reduction in CCN1 incorporation. So well, that was exciting to us because it suggested that this cell-based approach, where we took these primary cells from donors, made a matrix, actually pointed to trends in matrix composition and matrix function that seemed to align with what we saw from tissue. So that was very exciting, and we also stained for CCN1 in the tissues here, and we saw that indeed on the bone marrow side here, there was a lot less CCN1 than in the elderly than the young. What was interesting actually is when we stained muscle tissue in the elderly mice, we saw plenty of CCN1. So if there's a reduction in this protein, it seems to be more localized within the bone marrow niche, and not present in other tissues, because CCN1 is present in a host of other tissues in the body. It's really everywhere. So you know, it was important to also think about, you know, what site we're looking at. And with that, I just want to, you know, thank you for your time, and yeah, I'm happy to take any questions at the end.
Thank you, Milos, for such great work. We will now move on to our third speaker of the day, and this is Daniel Sloseris from Simon Fraser University, and we'll continue with questions at the end of the webinar. So Daniel is currently completing his undergraduate degree under the supervision of Professor Nancy Ford, where he's studying the aging of collagen, and Food Lab's biophysics research group investigating the molecular level physical properties of our body's building blocks. And today, Daniel will present his work entitled Aging of Collagen, the Effects of Glycation on Collagen Stability, Mechanics and Assembly. Over to you, Daniel.
Hello, thank you. Thank you very much for having me. That's right. My name is Daniel, and you know, we've seen a lot of very interesting information today about all the different things that's going on as we age, and we've talked about collagen. And I'm really going to focus specifically on collagen, and we're going to focus on one of the processes that occur as we age, which is glycation. Just before we get started, this is the campus in Burnaby, BC, British Columbia, Vancouver, where I'm coming from, just to get context. So, you know, we've seen aging is really quite complex, and I always like to bring up this idea of the hallmarks of aging, which is this idea that's been popular recently, a lot of these processes that sum up to create aging, and a lot of them have gotten a lot of attention, such as shortening of telomeres, cellular senescence, loss of proteostasis, as we've seen there's big changes in that case. And if you're here at this talk, you're probably interested in the molecular effects, or sort of what's going on at the molecular level. And now, you know, the hallmarks of aging encompass a bunch of different reactions and processes, but it's not fully encompassing. There's tons of things going on. And specifically, what I focused on is a reaction called glycation, which is essentially just the progression of the Maillard reaction in the body. And that's the same reaction that occurs when you cook meat, when you cook bread. It gives it color, it gives it, say, flavor, it gives it scent. So basically, at its core, it's just a reaction between proteins and reducing sugars such as glucose or ribose, primarily glucose in the body. And after a few steps, you end up with these irreversible products called AGEs, or AGEs, which stand for advanced glycation end products. So I don't know what just happened. Yeah, so AGEs, advanced glycation end products. And so this last step where they become these AGEs is irreversible. So these guys tend to accumulate in the body, and specifically in proteins with long turnovers, or short, very long half-lives. And as we saw from our first talk, collagen has quite a long half-life. And so these AGEs have two main detrimental pathways and how they affect us physiologically. One is through their interaction with receptors for AGEs, or RAGEs, And we know that these can cause all kinds of signaling that lead to oxidative stress, inflammation, whatnot. But on the other hand, they also cause structural or mechanical damage in the sense that they are a physical thing that's getting in the way, sticking onto proteins, sticking proteins together. And we know this leads to all kinds of detrimental effects, specifically with collagen in terms of your skin, your cartilage, your tendons, your bones, all these tissues that are very, very high in collagen. And also, glycation is thought to be one of the main drivers behind diabetes type 2 complications, due to the fact that in hyperglycemia you have way more sugar around. All this sugar is open to glycate your proteins, and so if you have more sugar, you have more glycation, you glycate faster, and so all these effects are just accelerated. Now, collagen, as we've heard a lot about today, I think is important to think about it hierarchically. So in the body, we see it in the form of fibrils, especially collagen 1, which is what I'm talking about today. And in the form of fibrils, there are fibers. It provides elasticity, stability, support. Cells interact with it. But really what we were focused on is sort of this bottom level. So the individual unit of collagen, or the collagen molecule, as I'm going to refer to it. And so the collagen molecule itself, if you haven't given it much thought, really is quite small and very long with respect to its width. So it's about 300 nanometers long, 1.5 nanometers in diameter. Quite a large protein as far as proteins go, but small when compared to things like microtubules in terms of its width. So it's a small unit at its base. And on the right here, we can see sort of a more accurate depiction of what these molecules look like. They have an overall right-handed triple helical structure with individual alpha chains that have a left-handed chirality. And so collagen, in the same sense of any protein, can become glycated. And so by the same reaction that I talked about before, we can end up with what we call glycated collagen. And so here I've drawn it as a triple helical structure that's slightly messed up. It has these ages that are attached on in different forms, maybe between chains, maybe just on one of the chains. And here are just some of the two examples of common ages, pentosidine and carboxymethyl lysine. And there are dozens of these, and these are just a few. We haven't characterized all of them. And I have the structure drawn a little wonky, but really we don't know exactly what's happening. This is sort of an interpretation of what we think could happen. And so the driving question behind this research was, how does glycation affect collagen's structure, its stability, its mechanics, and also its ability to assemble into those higher order structures, as we saw before? So to do this, we first induced glycation by incubating collagen with ribose. And we can use specific fluorescent wavelengths to track the progression of the reaction, because we know that some of these products that form, for example, pentosidine, as I showed on the last slide, fluoresces at a specific wavelength pair. And so we can use the accumulation of that fluorescent signal to track the progress of the reaction. And so here we saw, as we increased the amount of ribose at a certain amount of time, we had higher presence of these ages, confirming that something was going on. We also use SDS-PAGE to show that these individual collagen molecules were getting heavier. Their alpha chains were increasing in, or I guess decreasing in electrophoretic mobility. You can interpret that as increasing in mass, or in its ability, or losing its ability to migrate as far. And so we took this also as evidence, as modification of these proteins. We're changing them at their molecular level. And so next we asked about its stability, right? And so one way we can do that is using a technique called circular dichroism spectroscopy, which is a form of absorption spectroscopy where we shine right-handed and left-handed polarized light at collagen. And because of that right-handed and left-handed triple helical structure that I was talking about, you get this very specific spectra. And this is a characteristic spectra for collagen, where you have a positive peak at the roughly 222 nanometers, which corresponds to that right-handed triple helical structure, and then a big negative peak at around 190 nanometers, which is because of the individual left-handedness of all those alpha chains. And so here's what we found when we compared the glycated and the non-glycated collagen. We saw quite a small difference between their structure, so no real big change in the structure of this collagen. We weren't losing that triple helicalness to any measurable extent, at least not any significant extent as we would have expected. But what we then did is you can take collagen and melt it by increasing the temperature. And one way of tracking that is to use that triple helical signal at 222 nanometers. So if you just track that signal as you increase temperature, you can effectively track the melting. And what we saw here is that the glycated collagen actually melted sooner. So we had some thermal stability loss by glycation. So we found, these collagen molecules they're melting a little bit sooner. They're not as stable as their non-glycated counterparts. Another way of addressing this question that we thought of was trying to digest it with enzymes, specifically trypsin. And so we've seen, I think one of the previous talks mentioned that there is increased resistance to degradation, and this has been seen many times at the fibril level, but we were curious about what happens at the molecular level. And what we found here, interestingly, was actually the opposite of a lot of the work that's been seen, which is that as these collagen molecules were more glycated, they were easier to chop up with trypsin. We had more digestion of the more glycated molecules with trypsin. So this sort of supported that previous finding with the circular dichroism, where we had a loss of stability, which seemed to also relate to an increased susceptibility to digestion. Next, we addressed the mechanics, and to do this, we use a technique called atomic force microscopy. And so this allows us to take images of individual collagen molecules. As I mentioned, they're really quite small, so you have to use some sort of EM imaging or AFM, for example, which is a very practical way of looking at collagen. And so we can get these topographical images of collagen, which we can then trace with some in-house software, and then we fit to a model called the worm-like chain model, which just basically allows us to extract mechanical parameters like stiffness. And we do that through something called the persistence length, which is basically just a measure of flexibility. If you have a larger persistence length, you are less flexible, and if you have a lower persistence length, you are more flexible. And so we did this analysis with our glycated and non-glycated molecules, and what we found was that these glycated molecules were actually stiffer. So they had a larger persistence length, which was an interesting result considering the fact that we had previously seen that they were less stable. So in some sense, they were less stable, but also mechanically stiffer. So that raised some questions that we had to think about later in wrapping this project up into a paper. But finally, what we wanted to think about was, well, we have these collagen molecules that have been modified to some extent. We know they're changed in some way. Can they do their intrinsic physiological ability of self-assembly? And so in the body, what that is the self-assembly of these individual molecules into fibrils, which and then into fibers. And so this is very organized, very well documented. You can take images on the AFM of these guys, and they're perfectly debanded. They have that nice structure. And so this is what we would expect. One method of looking at it we found was with DIC, where we could visually and very easily see the formation of fibrils from soluble collagen into these fibrils, which are now visible. And what we found was that the glycated molecules just could not do it. There was no assembly at all after, the same conditions as we would have done with the non-glycated collagen, where we see assembly, we see structure, we just saw absolutely nothing. So we were sort of losing this innate ability of self-assembly due to the fact that we were glycating. And so this was also sort of an interesting result because we have, these less stable, more stiff molecules, and then in some sense, they are now sort of useless to us. They can't assemble, they can't perform their physiological task. So to summarize, glycation is a consequence of aging, and it just occurs in the body. It's non-enzymatic. There's not much we can really do to stop it. And we know it can modify the properties and functions of proteins such as collagen. We use STS-PAGE and fluorescence to track it very conveniently. And what we found was that these glycated collagens were less thermally stable, less torsionally stable is how we interpreted the increased susceptibility to trypsin degradation. We found that they were mechanically stiffer, and we found that they were unable to self-assemble. And with that, I just want to thank these people that made the project possible and really contributed a lot to it. And there's a QR code here to the paper itself, and I'd be happy to take any questions.
Thank you, Daniel, for your lovely talk. We will now welcome questions for any of the speakers. So if you have a question, please type it in the question box and shown in the image. And all the speakers now have a bit of a chat about their work and about the methods that they've used to address the key questions in ECM aging research. So I've got a couple of questions already. So I'm going to start with a question for Anna. So in this question, the speaker is asking, have you looked at other ECM proteins and other tissues and organs, Anna, given that proteomics is quite expensive approach? So have you utilized this methodology for other organs and proteins?
So yeah, it's expensive, but the nice thing about it is that the Silac food is obviously labels every tissue in the same kind of way. So you have, that's also an expensive part. So the fact that we can do that all in one go is nice. And so, yeah, we have started to look at a couple of other tissues. Looked at the kidney and the heart and the brain.
Yes, and so what would you be expecting in those other organs? Are you expecting similar changes, or do you think there will be a difference between these circadian and non-circadian pools in other organs?
Yeah, so our kind of hypothesis is that it exists in the tendon because of this mechanical activity that is rhythmic, and perhaps in a tissue like the brain, they don't have that same mechanical demand in a rhythmic way, and so we're expecting to not really see these separation between these two pools in the brain, but perhaps we would in the heart because that has this more mechanical rhythmicity, and that is those things. So that's nice.
And are you planning to look into any other mechanisms or pathways of potential proteins that are part of, for example, this more dynamic pool, and how does that actually support the mechanical properties of tendons with aging?
I've actually now left the lab and moved to a postdoc, so but hopefully they will do in Manchester. Yeah.
Okay, thank you. So the next question is actually for Milos. So Milos, the question for you is how long you have counted MSCs when harvesting cell-free ECM? If you could maybe give a little bit more know-how about your methodology there.
Sorry, Vanja, you were cutting out a little bit towards the beginning. Did you say how long are they cultured before we make the ECM?
Yes, thank you.
Oh, okay. Yeah, it really depends on the seeding density. So currently we're culturing for about a week, but it really depends, I think, more on seeding density than anything else, because when the cells get overconfluent, they stop making matrix, and you actually do induce with ascorbic acid. So they're induced to make ascorbic acid, and when you induce them, they'll start to make matrix, I think, no matter when you induce them. So if you induce them too early, you're going to get a very patchy, poorly adhesive matrix, whereas poorly cohesive, I should say. If you do it too late, I think then you get a matrix that detaches or is too thick. So it's good to sort of induce them at around 75% confluence and have that be your standard confluence and not time.
Okay, that's brilliant. Thank you, Milos. So I think I have here one more question for you. Have you actually looked at CCN2 and CR61? Both of these proteins increase in fibrosis, and how does that relate to aging?
CCN2 and CR61?
Yeah.
So yeah, so CR61 is CCN1, CCN2 is CTGF, and yes, they are both actually. It's interesting. The number one protein that the number one by composition protein in the young matrix was CCN1. The number two was CCN2. So they were both in the young matrix in our data set and not in the aging. But on the other hand, they're involved in fibrosis. The thing is, I think CCN proteins are involved with new tissue formation across the board, whether that is an ECM and matrix deposition across the board. So CCN1 is embryonically lethal, for example. It's very important in development. It's not solely related to fibrosis. Now, it's absolutely involved in fibrosis insofar as fibrosis involves matrix production, which it certainly does. I mean, I think that there's kind of a changing thought about fibrosis in general, which is that fibrosis is just another type of extracellular matrix. Essentially, it's just a dysregulated one. I hope that answers your question.
Yeah, thank you.
I think so.
Yeah. So the speaker, the attendee is interested in the use of proteomics to investigate predisposition to arthritis in the extracellular matrix. So they're interested to know, would there be a place to assay CCM protein in that context?
Yes, absolutely. Although I would say that, you know, CCN is just one player, and there's many. And I definitely don't want to create the impression that this one matrix protein is in charge of everything. Explains it's far from it. I mean, it really couldn't be further than that. But it's important, and I would say, yeah, definitely look at CCNs. But there's all sorts of other proteins that are involved too. I would say to first look at a matrix produced by healthy cartilage-derived stem cells. There was a publication in, I want to say it was in Biomaterials. It was from the New Jersey Center of Biomaterials. It was by Young Mao a few years ago, where they did just that. They looked at a chondrocyte-derived matrix, and they did proteomics. So you get a baseline of what the chondrocyte matrix sort of looks like. And then from there, you could look at, well, what happens during arthritis? That would be my thought initially.
Brilliant, thank you, Milos. The next question is, so from the attendee, they're saying collagen in the body is acted on by collagenase. So have you guys tried degradation profile by collagenase? So I guess this could be either for Anna or Milos.
Oh, I haven't done that.
I think that might be for me.
Or maybe for you, Daniel, yes?
I was going to say.
Yeah. Yeah, so there's tons of proteins that chop up collagen in the body. That's true. Like MMPs are a great example. We used trypsin, but we also wanted to use other ones as well. There was no real like, okay, we're going to try all of them, but it would be great experimentally to do as well, to test other proteins as well. We work with bacterial collagenase, we work with MMPs as well, but it just didn't go into that specific project.
Thank you, Daniel. So one more question for Milos. So were you looking at all at the CCN1 in the muscle tissue of aging mice? And could you see the levels more elevated in the young? And if so, is there a response to lower CCN in the bone marrow? So could there be a change in the muscle tissue, and that's kind of reflecting on changes in the bone, I guess?
I think that's certainly possible. And we really didn't carefully look at muscle tissue. It was really just kind of an observation that we made along the way because our tissue sections had some muscle in them. But we pretty consistently saw, I would say, similar levels. I can't say that it was higher or lower, but I would say similar levels in the muscle. So, but I think it's definitely worth looking. I mean, CCN1 is absolutely found in muscle, and we really don't know what the role in aging is.
Thank you. And the follow-up question is, could you give some explanation as to why the matrix in young bone cells is stiffer? Could it be that young cells produce more enzymatic cross-links?
I mean, again, I think that's possible, but also, I mean, if you look at these cells in culture, and this has been published on pretty extensively, young cells are also just physically smaller than the aging ones. Usually aging MSCs actually get larger, and you have, and I'm sorry, this is maybe an oversimplistic explanation. I think Daniel probably, his work could really shed light on this question a bit more, as well as Anna's with composition, but they're bigger cells. And I think that just physically, their shape is much, you know, their circularity, their spreading morphology looks very different. And actually, I think that there is a effect of cell shape, size, spreading morphology on the matrix that they make. And I think that that's the starting point. Are there different, is there different processing of those matrix proteins, cross-linking? I'm sure there is. But the first place I think of actually the cells that are actually making it, because I can see how they're making it before I decellularize. And you know, the aging cells are just much larger, much more varied in size, spreading behavior, and the matrix really reflects that. You know, interestingly, the young cells, they tend to grow in much more like parallel organization. And when you decellularize it, you'll see the same sort of directions in the matrix that they leave behind. So I don't know, that's food for thought, but just something to keep in mind.
Thank you, Milos. So a question for Anna. Was there any other CSPGs, Anna, that were upregulated or downregulated between the aging and the young tendon tissue that you looked at?
There were changes in some glycoproteins and proteoglycans, yes, but I can't tell you about the specifics, I'm afraid. It's been a while.
Okay, thank you, Anna. And now for Daniel. So Daniel, the question for you is, so is collagen being glycated in the aging process across different tissues? And if so, are they glycated to different degrees? And is there a typical phenotype for all of them?
That's a good question. So as far as we know, glycation is spread. So like it is everywhere. I mean, everywhere you have sugars, because that can interact with proteins, in theory, you should have glycation. And in theory, like the rate shouldn't vary, but what does vary is the turnover of proteins. And Anna talked a lot about this, and it was great to bring relevance to what I was going to say, because it's more so dependent on that turnover. If they turn over quick, the accumulation of glycation is not very significant because it really is quite slow. I'm talking like years. So if these tissues turn over quick enough, you don't really see accumulation. And that's why sort of the work is focused on proteins like collagen that do stick around for so long that it's enough to actually have a detrimental effect. And I think a good way to think about it is like when you go get your A1C, at least that's what we call it here, your blood sugar tested. When they look at your average blood sugar over the last 3 months, basically what they're looking at is how glycated is your hemoglobin. And so hemoglobin turns over about every 3 months. And so when they look at it, they can get an idea of how much sugar was in your body for the last 3 months. So that's sort of contextualize that sort of idea.
I've got a couple of more questions for you, Dan. Thank you for that. So one question is, as you use the ribose sugar in your experiments for glycation, could you use glucose, and would there be a reproducibility of results?
Yeah, so there's been a lot of work published on this. Ribose has been deemed to be quite an effective replicate, essentially, of glycation. You do end up seeing a lot of similar products. For example, the pentosidine that I showed, and this has been shown with mass spec, not just fluorescence. But really the main difference, and the reason we use it and a lot of other labs use it, if you look into it, you'll see a lot of papers do use ribose because it's much faster. I've done testing myself as well, like glucose is just way slower. And so if we're doing things at 37 degrees, I mean, these could be experiments that to get to an appreciable level of glycation to be like a month, maybe. And so, you know, it depends really on your time frame if you want to accelerate, if you're willing to spend that much time. But yes, obviously, like, our bodies don't have that much ribose, and I think it's important to note that, like, my experiments were not with the physiological range of ribose at all, but we were just sort of trying to limit test to see what happens at the limit of this if you have tons of sugar around. But yeah, it is more relevant to think about glucose. But there's other sugars, something called methyl glyoxyl, that apparently is a very powerful glycating agent that also exists in the body. So there's a lot going on.
Thank you, Daniel. One other question for you as well. So if glycated collagen could not self-assemble, does this mean that proteins that become glycated in vivo would also not be able to remain assembled and would break down as aging continues?
That's a good question. I think if I understood that correctly, is does the glycated collagen as we get older affect our ability to regenerate tissues? I think that's what I understood.
Yes.
Yeah. So that's a great question, and I think what's important to note as well is in the fact that this study was non-physiological in the sense that in your body, collagen is secreted and quickly formed into fibrils and incorporated into the extracellular matrix. So you don't really have free collagens floating around in the sense that I did during my experiments. And the reason we did this was to isolate the effects, because as soon as you have a fibril, you're kind of protecting those interior collagens from the sugars that are in solution, and so it's not the same effect. And a lot of work has been done on those types of systems where we have a fibril, you add sugar, it does something to the fibril. But we wanted to know, you know, what happens to the individual molecule. So I think it's not easy to draw direct correlations to physiology from the results that I showed, but it's more so interesting to think that these effects happen to every single molecule that make up a fibril, or could happen, at least to the exterior, the ones that are exposed to ribose and glucose.
Yes, thank you for that. So I haven't any other questions in the box. So please, for the last couple of minutes, if anybody has any other questions, please type them in a chat box. I have just a quick one or two questions. So one for Milos. So Milos, I found this result that you showed with CCN1 recombinant proteins fascinating, that the cells were able to restore their responsiveness to various growth factors and also like differentiation induction. So, so what do you think about therapeutic potential of CCN1? And are there any, for example, lifestyle interventions that are known to increase CCN1, like diet or exercise? Is there anything that we could do to increase CCN1 in our bodies, basically?
I think it's a great question, especially about diet and exercise. And I am really looking for collaborators to send me bone tissue. From their studies, we can look at it and assay some of these matrix components. I have one, but I would love to work with anyone and everyone, really. But I think that's a great question, because if there are, lifestyle changes that could keep a healthier microenvironment or bone marrow niche, and we could sort of measure that and analyze it, then that's fantastic. I think from a therapeutic perspective, there's different approaches. There's gene therapy to increase the expression of these things and kind of remodel the matrix, or there's other methods. I can say we're working on something that's actually related to trying to supply the recombinant protein directly to the tissue. So there's definitely different approaches to do it. But I think it's I hope it's an interesting idea, this, different matrix components and using that as an approach to, restore tissue function. So.
Yes, thank you very much for that, Milos. So I haven't got any other questions in the chat box. Do speakers have any questions for each other?
Actually, I had a question for Anna.
Carry on.
Hi, I was curious. So the SILAC, because that's something that, it's a fantastic approach for precise quantification of the matrix components. But do you know, because it's a physical, it's a physical additive to these matrices, essentially, to the proteins. So do you know that the SILAC, does the SILAC process itself alter matrix organization? Or basically, if we did this, if we took this approach and used it on the matrix as we produce it in my lab, would it alter the product that we have because we use the SILAC?
I haven't looked extensively into all of those things, but as far as we can tell, it doesn't affect it. So we also had kind of matched control mice that were fed exactly the same diet, but with 12 carbon rather than 13, and like the abundance of all the matrix proteins correlate really well between the two. They can't see any difference at like that level. We haven't looked at a kind of more structural level, but I think the difference is probably small enough that you wouldn't really see. It's just carbon atoms rather than a kind of big tag or anything like that.
Very cool. Thank you.
Oh, thank you for that, Anna. And I have one final question for Daniel as well here. So Daniel, would you just be able to tell us how and why you think glycation decreased the thermal stability of collagen?
That is an excellent question. We speculated a lot when we were trying to wrap up this project and trying to think about possible reasons for not just that result, but the other ones as well. We're still quite perplexed about it. As far as the stability result goes, we know, well, we think of denaturation as the loss of triple helicity, so sort of this unwinding. And what we're thinking is that maybe like steric interactions, these groups that are getting added on potentially could be like pushing the individual chains slightly further apart than they are otherwise. Somehow they're bringing this tightly coiled triple helix into a slightly less coiled triple helix that can then melt easier or disassemble more easily. So we think at the base of it, our idea right now is sterics, is like a steric effect. just, you know, having things in the way that push chains out or shift chains around. But it really is quite a difficult thing to reason with, especially when you consider like the fact that we saw that they were stiffer. So like, you know, how's that consistent? And so, yeah, we have a lot of questions left. It's definitely not a fully, like, the work is there, it's been published, but we don't have a great way of explaining what we saw just yet.
Well, thank you very much for that. And I think I will leave it with there for questions. I think that's been a great range of talks and great range of methodologies. I'm really impressed with the range of methodologies that you guys used and shared with us today. And I hope that our attendees can get in touch with you if they need to collaborate and use any of these brilliant technologies that you've used. We hopefully will also continue this conversation further online. So please follow us at Biochemical on Blue Sky and X. We are also currently 2025 ECR webinar series. If you've been inspired by any of the speakers today and would like the same opportunity to present your research, then please consider submitting an abstract for consideration by 24th of February and visit our website for more details. All of our upcoming webinars are listed on our website. And if you missed any of the 90+ webinars that we've already run as part of the series, or you would like to watch them again, please visit us on our website or our YouTube channel. And the recording from today's webinar will be available to watch within the next couple of hours, or a couple of weeks rather. Also join us please on 12th of February at 2 o'clock for our next webinar, which will be titled From Postdoc to Group Leader, Starting Your Own Lab, where our speakers will be discussing their experiences and providing advice on both UK and international opportunities. And finally, I would just like to highlight that it's more important than ever to stay connected and engaged with our fellow molecular bioscientists. So we encourage you to join the Biochemical Society's community of researchers and specialists if you're not a member already, and please take advantage of many benefits, including discounted registration fees for society courses and meetings, access to a wide range of grants and bursaries, summer studentship, personal online access to 2 of their journals, and much more. And during my career, I've been a member of Biochemical Society for nearly 15 years now and have reaped many benefits, including support for attendance at national and international conferences, stay connected bursaries for childcare support, and also taking active role in the societies as part of the committee membership. So please visit our website to find out more and engage in all these opportunities. So just once more, I want to thank all the speakers for a great webinar, and please have a great rest of the afternoon. Thank you.
Thanks for having us.
Thank you for having us.
Bye.